In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum, my dear students. How are you doing? I hope you all are well. This is Pakistan International School Types Virtual Learning System, session 2020. And this is the class of subject Science for grade 6. And today's lecture number is 13. Dear students, we are on chapter number 5, in which we are studying atoms, molecules, mixtures, and compounds. And today's topic is separating mixtures. If you see, this is our chapter number 5. And let's have a review of the previous topics which we have studied in this chapter so that you can revise all the topics which are related to today's topic. So first of all, in this chapter, we have studied about atoms and molecule, and we have seen some common elements and their symbols. If we just talk about we have seen some of the types of elements that are metals and non-metals and their physical properties and their uses. We have seen different compounds and mixtures as well, and their examples, and as well as the use of compounds and mixtures in our daily lives. And we have seen that air is a mixture of gases, and we have just talked a little about carbon dioxide, that how its level is maintained in the nature. My dear students, we will see today about that how we can separate mixtures. We know that mixtures are having some components which can be easily separated, and this separation is very much important in our daily life because it is having great application in our daily life, in great uses. So because that mixture is having different components, and these components can be separated easily as compared to compounds. Compounds are having such type of molecules and atoms in that which we cannot separate easily because they are chemically combined. But here in the mixtures, the components are not chemically combined, so we can separate them easily. So there are certain methods which we can adopt in our daily life as well as we use them in our daily life. We will see some examples also, and we will see that in the lab how can we separate the components of the mixtures. So first of all, or first and foremost, uh, the way or the topic related to the separating mixtures, or what type of method we can adopt, that's very, very easy, and that is having great application in our daily life. That is what? That is filtration. So filtration, what type of method it is? It is the method in which we use filter paper. That's why we say it filtration, because here we use filter paper or filter cloth to separate insoluble solids. The solids which are not dissolved properly in the water or in any type of liquid. For example, we try to dissolve sand in the water or soil in the water, but actually it does not dissolve, it just mixes with it. And when we keep that container for certain period of time, so what happened? Water will remain, make layers on the upper side, and the sand will settle down. So For the separation of insoluble solids from the liquid, we can use this filtration process. This is having an example in our daily life that when we strain our tea and the tea leaves left on the strainer, so that means a tea leaf does not dissolve, just they leave uh, the colors, the blackish and brownish color in the tea. The tea leaves doesn't don't dissolve properly in that water. This process is called filtration, that we are going to filter the tea. So we are having another example that water filtration plants are used in our daily lives, like by which we can separate solid impurities. Impurities means which make the water impure, dirty. And like with the water coolers in our kitchens, we are having filters just attached to it to clean the water. And after some time we see that that filter paper gets dirty, blackish or greenish in color due to the accumulation of lots of impurities in it. So regarding the lab activity, we can see later on after some time. But right now we can see some example that filtration of tea, like you can see here, tea leaves just left on, and this is called sieve or strainer. So this act as a filter paper or act as a membrane by which the water gets down and just tea leaves remain up on this strainer. This is very common example of filtration. And if we see, there are filtration plants, water filtration plants are installed in around the world, with the industries as well, where we can filter and we can clean the water. Some water evaporates here and there is a membrane by which the clean water comes here and then moves into the other places. So if we see how can we do in laboratory filtration, so you can see these are some apparatus which we can use for this purpose. So you see this is a conical flask, and this is also conical flask. This is a cone, and on this you can see this white paper, that is filter paper, and which is used here just to filter this chemical, or what we can say, any type of liquid which is having some insoluble dissolved substances. So this is in laboratory filtration process. You can see here is a beaker, or which is having muddy water, and when just we strain and we pass that water through, this is a filter paper, we pass this through filter paper, so water is coming down here, and we can see that this residue, or this is called mud, remains up in this filter paper. You can see here properly. So where we can find filtration in our daily life, not in daily life but in our body, there are lots of filtration processes are present. So first of all, very common, we can see the hair in the nose. If we see, the hair and mucus present in the nose help in the filtration of air before entering the lungs. Because if this like the air which we breathe, it just goes directly into our lungs, so our lungs will be accumulated with that dirt and that germs, so we can ill very soon, we can be sick. But in the nose from which we breathe, there is a mucus, Mucus is a sticky substance. You know about this in the previous classes you have studied. And some cilia. Cilia are the hair-like projections. Hair-like substances are present in our nose. So they propels continuously by breathing of the air. Breathing is in this direction. If you see this arrow, so air comes here at this direction, and you see the mucus is also coming in this direction, and due to this these cilia, that mucus is always in constant motion, and that it is used to accumulate or to stick the dirty material, dust, germs, smoke, and etc. And the air which goes in our lungs, that becomes clean and a little bit warmer according to the body temperature. So number two example is very common, that is in our kidneys. In our kidney, you can see this is our kidney. So in our kidney, these are millions of these type of systems present. These are microsystems, like very small, which you can see through microscope only, and they are called nephrons. So if we see not neurons, neurons present in the brain, but here nephrons. So you see from here, this is the part of the nephron in which blood from our body enters here through this artery. And what happened, with the pressure of this blood, all the waste material which is present in our blood just comes in these tubes, and from these tubes, that water or that substances including water, that uh, is changed or transformed in the form of urine, and this urine is removed out. All this all is having these type of systems present here, and then these are tubes, and urine is, comes here into the ureter, and then it removes out of our body, in which there are lots of waste material. So filtration occur here in this cup shaped part, that is called Bowman's capsule. If we see the next common example in our daily life, that is the use of water filters at our home, with the taps in the kitchens, which is like by which you are and like always available with the clean water. There are three tanks in which there is filter papers, and these filter papers filters lots of materials, and like the germs, bacteria, algae, and some minerals also which come in the water, and then you get clean water from the running tap for drinking purpose. This is also an example of that. Next way or next process, next method by which we can clean or separate the mixture, that is sublimation. Sublimation is a process, is very special process, that in which a solid on heating, when we heat the solid, it directly changes into gas form. Isn't it a different process? Yes, definitely, because most of the solids, when they heat it up, they just changes into which form? Liquid form. Yes, liquid form, and then this liquid changes into what? Vapors. Like you can take an example of water. Solid is ice, and water is liquid. So ice changes into water, and then water changes into steam, into the gas form, and like that vapors form. But here, solid on heating directly changes into water vapor, sorry, not water, vapors of that substance. And when we cool down that vapors, that directly changes into solid, not the central process which is changing into the liquid, that is skipped here. And that is a chemical reaction. sorry, that is not the chemical, it's the physical reaction, because solid changes into vapors, and vapors on cooling that directly changes into solid. So this is an activity which is done in the laboratory. That is an easy process that can be used an example. So here we go, we can see this example here as well. So this is, if you see, this is ice, it melts and changes into water, and then it, on giving more heat, it just evaporate, and that changes into steam. And when we cool down the steam, it changes into water, and then water, when we cool down more, it changes into ice in the freezer. This is a common process. But what is sublimation? When a solid directly changes into steam, and steam on condensation, condensation means on cooling, directly changes into desublimation, is done at that time, like changes into solid. If you see in this equation, this is a common process, melting of ice into liquid and then into vaporization, like vapors are formed, that is called evaporation. And on cooling, that it becomes again water, and then again freezing, and solid, uh, they form into the solid, that is called condensation, or like cooling of that water vapors, and that is called freezing, when the liquid changes into solid form. But here, that is sublimation, when solid directly changes into gas form, and gas directly changes into solid form on deposition, like on cooling, and that is called desublimation as well. Here we go. We are having a very interesting example. This is a solid iodine. Solid iodine, you can see on heating in the lab, we are doing this one, just a little quantity of solid iodine, and we're giving heat to this, and then it is changing directly into vapors. Look at this. Yes, it glows, it blows, and it changes into vapors. That is very interesting. But it is also dangerous. If you see this one, this is very common example, that is dry ice sublimation. Dry ice is solid carbon dioxide. Carbon dioxide in the gas form, it is not like this, but when carbon dioxide gets cooled down and changes into solid form, it directly changes into carbon dioxide gas. So you can see here an example of that, dry ice sublimation. So here is the laboratory experiment, and you can see the change of state directly from solid to gas and from gas to solid, that is sublimation. And a solid camphor or ammonium chloride, when it is heated, so it directly changes into like here is cotton, so that water vapors does not escape. So you can see here on both of the sides, that gets solidified as well again. So this is china dish. This is a small dish which is used in the lab. This is burner. This is cone which we keep on the to cover this china dish. And this is inverted funnel, we can also say there's funnel. So you can see it's this is like cartonic diagram, and this is original diagram, like this is china dish, and you can see these white drops, or you can see white crystals like structures, that shows you that this camphor later on just changes into solid form on condensation. So here are some examples of sublimation. That is dry ice, which we have seen before. Solid iodine we have seen before. Ammonium chloride, that is camphor and naphthalene balls, which we use for daily purposes. Like solid iodine, you can see that changes directly into iodine vapors. Camphor is solid on heating, that changes into gas directly. And if you see, this is dry ice. that solid carbon dioxide directly changes into gaseous form, and as well as this is mothball, or this is called naphthalene balls also. And naphthalene, or that is its chemical name, p-dichlorobenzene. So that is these are the balls which we used for insect repellents or kill the clothes moths. We just keep them in the clothes. So when we store the clothes, we store something for a longer period of time, we use them in washroom. They are used as insecticides. Yes. Okay, this is the next uh, procedure by which we can separate the mixture, that is distillation. So what is distillation actually? Distillation is the method by which two or more liquids in solution are separated by boiling of the liquid or with the lower boiling point and condensing into another container. It means distillation is a process in which we use mixture of the liquids to separate them out. This is laboratory apparatus. All this one, this is round bottom flask, this is conical flask, this is a tube, that is a distillation tube. And from here what happened by heating, okay, here very important thing is this one, the liquid which is having low boiling point, that boils first, and then it just changes into vapors form. And then when it moves from here, look here in the center there is a tube, from here that it changes into liquid again, and then it is collected here in this flask. Here there is a water outlet and inlet, that is for cold water. Cold water is running around this tube to just change this gas into liquid form. If we see this process, distillation in the laboratory, how it's done, so you can see this process. It's so interesting, no? There's a lot of apparatus setup is a little bit harder, but you can see here the mixture of liquids is boiling. So you can see here what vapors are formed, and from here when this is going on from this tube, here the water is inlet and outlet. So what happened, look here, the water is always present to cool down the temperature, and these vapors of the liquid just changes again into the liquid form. So the liquid which is having low boiling point, that uh, boiled up first and just collected here first. Then we change this container and the other liquid will come. Because here we can see the temperature at, and that we are having check and balance on that at regular period of time. So if you see in Saudi Arabia, in lots of countries, what happened, we use oil refineries and we use distillation process for that. Because that is this country is the big exporter of the oil. So what happened, we use lots of refineries are made in like Middle East, etc. So because the crude oil which comes from under the ground, that is refined here, like that is distilled here. And because crude oil is having lots of mixtures inside it, like it is a mixture of different petrol, diesel, kerosene, gasoline, etc. Lots of things are present in it. So all the material stuff is separated out here in these oil refineries. Okay, now we will see the next one, the fourth, last but not the least, that is paper chromatography. And in that we separate colors. Chroma means colors, graphy means that the procedure to make a graph, like to make a proper indications of the paper. So that is separation of colored chemicals. That is having great importance in the lab, as well as if we see the type of chromatography is paper chromatography, in which we use paper, filter paper, or some other type of paper, by which more soluble dyes moves faster in that. Like for example, we are having food colors or pen inks or any type of dye of the clothes. So the more suitable dyes move faster on the filter paper than less soluble dyes. So which dye is more soluble in the liquid, in the water, it moves faster. If you can see different colors in this. So now you can see, one drop of in different colors, because there's a mixture of different colors. So the which is more soluble, that runs more quickly to far away. So this is a laboratory experiment. You can see here, this is a filter paper, and filter paper is hanged in this water, that is a beaker in which we are having water, and you can see ink. This is blowing, so you can see different colors in that. So we can see separate colors in this. So here is an activity of chromatography which we do in the laboratory. That is simple one. A medicinal dropper with, this is a filter paper in which we put with the dropper a dye or ink, a drop of that, and this is a beaker in which we are having water. So we hang this paper in the water, so this dye, the most soluble color of that dye just moves more far away from that. You can see here also this one. And this is a filter paper, it's hung in the water, so you can see this one. So the colors are separating here. Yes, because we have discussed all the lab activities here. So here is some science safety rules as well, that are called lab safety rules. Otherwise, you can have such type of condition, God forbid. So if we see, when we go to the lab or science activity, just listen to your teacher first carefully. Follow the teacher's rules. And number two, don't put things into your mouth. To check any type of object like sugar, salt, or what type, or you don't know it is sugar, it is salt, or what type of chemical it is. Don't put that in your mouth. That is very, very harmful. That can burn your whole throat or your stomach, etc. That can be acid, or that can any type of other chemical which is dangerous for our health. Wash your hands after the whole activities, because maybe some chemical just stuck with your hands. Be careful with the sharp tools like scissors, like a scalpel, like razors are over there. So just be careful with them. Use goggles, proper clothing, lab coats are there, goggles are there, and you can have don't taste and smell materials, handle scissors and sharp things carefully, keep your place neat and clean, and your all the here instruments which are using, that must be cleaned or washed before the experiment. Otherwise you cannot get the proper result. And as well as if there is any problem, any unsafety over there, just talk to your teacher, and always practice science safety rules. So that's it, my dear students, for today. With this topic, we have completed our chapter number 5. Inshallah, we will see you in the next lecture, in the next chapter. And until, wish you good luck.